Greetings everyone and welcome to DDL 2020, which this year is a bit different to what we're used to. Today, Raquel from Hovion and myself, Anna Sipitanou from Copley Scientific, we will be exploring the potential and practicalities of semi-automation in inhaler testing. Just before we commence with the technical parts of our presentation, let me take a moment and introduce the two companies to you. Hovion is a contract development and manufacturing company dedicating to helping pharmaceutical customers bring a new and off-patent drugs to the market. Hovion has over 60 years of experience in the development and compliant manufacture of APIs and drug product intermediates. Copley Scientific, on the other hand, is recognized as the world's leading manufacturer of inhaler test equipment and prides itself for being a key customer's companion in their drug approval journey the past 74 years. The topic I will be touching on today is variability. And variability in inhaler testing is a very big discussion around the industry. The main two impacts of variability in new testing are the failure to achieve mass balance, which means that not the whole dose has been sized, and APSD with errors, which means that the dose is sized incorrectly. These lead to repetition of tests, low productivity, low morale of the personnel, inability to discriminate between product variability and other sources of variability and others. And the overall take-home point is the cost to the organization. And that's something every organization is trying to avoid. I've tried to summarize the different sources of variability. And one of the main sources is, as you would expect, the human factor, or else known as the analyst-to-analyst variability. Let's take a step back and imagine a laboratory setting where you have different analysts with different backgrounds and different experience. Every analyst will probably perceive the information in a different way, so they will interpret the method in a different way. Also, they will have different levels of training. Some of them they will be lacking training, some of them they will have sufficient training. And let's not fool ourselves. Running NZIs the whole day can be rather monotonous. But it's not only the human factor that increases variability. Methods are another great source of variability themselves. How well a method is written, how much detail does it include? Has it taken into account the system setup optimization? Has it accounted to site variances in the case of method transfer? All these questions and more have to be answered by a very well written and specific method. Last but not least comes the environmental variability, and we have to remember that environmental factors that can affect our testing have to be specified in the method and monitored. And as you can see, pretty much here everything is interlinked. Considering environmental factors will help you create a more specific method. A more specific and well-written method will help reduce analyst mistakes, thus reduce variability, and the circle continues. There are numerous pieces of equipment that you can use to improve reproducibility and productivity and reduce variability of your testing. But for the purposes of this presentation, we will be focusing on APSD recovery equipment. But let's see the different ways you can use to recover sample after an APSD test. First of all, we have the fully manual process. Here the analyst dispenses necessary amount of solvent in caps, induction ports and pre-separators, shakes manually, and samples with a pipette into HPLC vials. A step further towards more automated recovery is introducing the gentle rocker and SPU. Now here the analyst will still need to dispense specific amount of solvent in NZI caps, induction ports and pre-separators. However, the shaking process will happen through the gentle rocker and SPU at a constant speed for a specific amount of time. Then the analyst will still sample into HPLC vials manually. The optimal way for APSD sample recovery with increased reproducibility and productivity and reduced variability is by using a combination of the NZI system and the SPU. But let's see what exactly the NZI assistant does. So the NZI assistant is a labor-saving device that operates in 3 distinct steps. First of all, it will dispense a specific amount of solvent in each cap of the 3 NZI cap trays. Then it will gently agitate in order to dissolve the active drug in the solvent, And lastly, it's going to sample from each of the NGI caps into HPLC vials, preparing samples ready for HPLC analysis. Throughout this process, as you appreciate, the analyst is not very much involved, so it frees up analyst time, reduces intra and inter-analyst variability, and also it's a very safe and highly efficient way for your sample recovery. I'll pass it on now to Raquel, who will explain to you what happened in the study.
So, in this study, a mainly manual analysis was compared with more fully automated analysis, using this system to demonstrate two things. First, the time savings, and second, if the data from two setups are equivalent. And for that, aerodynamical particle size distribution data for a twin caps dry powder inhaler were generated using two different methods for drug recovery: an essentially manual recovery method, aided by an automated solution for agitation of the solvent in the NGI collection cup tray, the NGI Gentle Rocket, and we also used a fully automated recovery with an NGI assistant. All the tests were carried out using an in-house method developed, and drug recovery solutions were then quantified using an HPLC system, and the resulting data were analyzed using Empower software. CITA software was also used to generate aerodynamical particle size distribution metrics. In order to do the comparison study, 23 replicate tests were carried out in total: 10 by analyst 1 using the manual drug recovery method, 3 by a different analyst, a second analyst, using the same method, and 10 using the more automated method. The equivalency between the datasets was assessed via t-testing for determining the extent to which two datasets are identical, and this graph shows the percentage variance in the amount of drug recovered from each stage of the impactor for the runs carried out by analyst 1 alone and for the two analysts combined. Through these results, we can see that, in general, variability increases when measurements are carried out by multiple analysts, even with a very small number of runs performed by the second analyst. Usually, more than two operators are responsible for analysis, and for that reason, variance may reasonably be expected to be even higher. Moreover, the impact of the operator variability is likely to be higher with a completely manual method. Since it would be rare for analysis for a given product to always be carried out by a single analyst, the combined analyst 1 and 2 dataset was selected as the more realistic basis for comparison of the impact of switching to automated drug recovery. And this graph shows t-test data for a comparison of the results produced by the established, a more manual method, analyst 1 and 2, and via automated drug recovery. When automating a manual method, it is crucial to confirm that the results are equivalent using statistical methods such as these. And here, analysis shows that for every stage, p-value is well above the threshold value of 0.05, confirming statistical equivalence. Robust equivalence is also observed in the aerodynamical particle size distribution data parameters generated by each method, as we can see in Table 1. Through the monitoring, analyst bench time during the study enabled calculation of the productivity gains accessible by switching to the more automated methods. And as you can see through the comparison between both process results for 3 runs, we have a total amount of time spent of around 225 minutes for the manual process. On the other hand, the time spent for the same 3 runs on the automated process is only 140 minutes. And the results indicate that analyst bench time is reduced by around 40%, which is a substantial increase in productivity. The study also provides a good illustration of the potential to progressively adopt automation solutions, with cross-validation each stage, reducing the risk of having systematic differences. In a matter of conclusion, the semi-automation of cascade impaction has an important role to play in improving the quality orally inhaled products test data, while at the same time reducing the cost and effort associated with routine. This study illustrates how sophisticated, easy to use, off-the-shelf solutions can be used to generate statistically equivalent data and deliver major productivity gains. And by freeing analyst time for more valuable, less repetitive tasks, such solutions can help to minimize the risk of out of specification results, address health and safety concerns, and at the same time deliver an attractive return on investment. Thank you for your time. I hope you enjoyed the session.